Good evening, everyone. I'm Richard Harland, Dean of Biological Sciences at UC Berkeley. Welcome to you all to Season 12 of Basic Science Lights the Way. Thank you for joining us, whether this is your first visit or if you're a regular. We appreciate your interest in basic science research. I hope that this series will reinforce your sense of connection between fundamental science and the innovations that will shape the future. Tonight, Berkeley biologists will be discussing zoonoses, which are infectious diseases that have jumped to humans from other animal species. We're all familiar with the plagues and pandemics that have been a scourge in human history. Today, we will hear how disease spreads where people and animals interact, and how we can monitor and predict the spread of infections, and perhaps predict where they'll jump to human populations. Tonight's panel reflects Berkeley's expertise and world-class talent, and I'm excited to hear from three of our faculty on their current research on people, bats, and gorillas, and where the field is going. Now it's my pleasure to introduce our moderator and first speaker this evening, Professor and Department Chair of Integrative Biology, Mike Boots. Research in the Boots Lab focuses on the ecology, epidemiology, and evolution of infectious disease. He uses a combination of evolutionary theory, experimental host parasite systems, epidemiological models of wildlife and human disease, and field entomology. Mike, thanks for taking the reins tonight. I'm looking forward to where you take the conversation.
Thank you, Richard. It's great to be here with you all this evening, and I'm excited to be part of this conversation with my colleagues. My research is currently really driven by the fact that we don't have a good fundamental understanding of processes that lead to disease emergence in new populations. And clearly, as we all know, the COVID-19 pandemic has shown us just how devastating zoonoses can be. Before I expand on this a bit more, I'd like to remind you all that we're going to address your questions at the end of each talk. So please add those to the chat. We'd like to hear from you. Okay, so it's my pleasure to kick off tonight's event as the first speaker. Let me just share my screen. Okay, so as I said, I'm interested in the evolution of pathogens and of emerging pathogens. I use a combination of computer and mathematical models, and also some model systems. There's a lot of zoonoses out there. All of these are important zoonoses. We know that there's going to be more and there's going to be new ones, but we don't know when they're going to arise, and we don't know how bad they're going to be. To some extent, the state of the art could be that they emerge at random and then they do random things to us. Okay, clearly that's not true. And given what happened in COVID-19, we need to understand this a lot better. And that's where our research is driven. What I want to talk about is the evolution of infectious disease when it first emerged into humans. When it comes into a human, we're a new host, and therefore the pathogen really is just not adapted to us at all. And so when it first comes into us, it causes all this harm and it causes this virulence. And, that might just seem to be some sort of accident that happens initially as it comes into us. And lots of scientists said at the start of the SARS-CoV-2 pandemic that what's going to happen is it's going to start off being severe, but then it's going to adapt and it's going to become milder and less of a problem. And and this kind of makes sense, right? So if you're a pathogen and you kill your host quickly, once you've killed your host, you can't transmit anymore. A bit less dramatically, in the human context, if you make us ill, we'll stop walking around and contacting people, or we might isolate ourselves, or we might go to hospital and then we'll be isolated and we won't be able to transmit, right? So that high severity and that virulence is kind of shortening your infectious period, the time over which you can infect. And that's bad for the pathogen because what the pathogen wants to do, pathogen reproduction for the pathogen is moving from one host to the next. So evolution should be selecting against it. So maybe this trip comes in, this severe zoonosis that will start selecting against it. So that's why people were saying that. Did that happen? No, don't think it did happen at all. I grabbed this data and asked Gemini to make me this slide. And there's just no doubt that if you look at something like, you remember these variants that we had during COVID by 2021, that the alpha variant was way more severe, way more virulent than the initial COVID when it came into the population. As we go through the COVID-19 pandemic, our perception of how severe it is getting more difficult because lots of us have got infected and then have got immunity, or we've been vaccinated. And what the immunity does and the vaccine does, as well as reducing the chance that we're going to get COVID, it also makes it really reduces the severity. So there's a sense by the time I got COVID, it was the Delta variant. But I'd been vaccinated, so it felt quite mild to us. So it's then hard to know how virulent it really is. But without any doubt, COVID when it's running through the population, it's adapting to us. It's getting much better at transmitting human to human, right? But even Omicron was not mild. We were just all vaccinated. The tragic story with Omicron is if you go to places like Hong Kong, where people in care homes were quite well protected, but they hadn't been vaccinated and they had not, they're not been exposed at all. Omicron comes in, it transmits way better, and it gets into these care homes of these unvaccinated and non-immune individuals, and the fatality rate amongst them was really high, like 28%. So it's clear that we didn't just get this adaptation to becoming milder. So why is that? Why do we think that pathogens kill their host? Well, we think it's all to do with trade-offs, okay? So if you imagine within the host, you're a parasite and you're replicating within the host, it seems really likely that if you're replicating faster within the host, you're a strain that's really replicating quickly, you're producing more potential transmission stages, more viruses, more protozoa, whatever it is. So you can transmit faster, okay? That increased replication is likely to be causing more damage as well. So that's increasing virulence. That's causing the host to die quicker. So really what we think underlies this idea that you might end up at some sort of optimal replication rate is you're balancing how fast you can transmit relative to how long you transmit for. So do you go really fast and then can't transmit for very long because you kill the host, or do you transmit at a slower rate for a longer time? And there's some optimal balance between those two things. And that's a general idea of what we think might be happening. Is it true? We've got lots of data that says this probably is true. In animals, we can do experiments. This is a great example of it. It's Marek's disease in chickens. There's other examples as well, very virulent disease in chickens. And you can see in this graph, there's some sort of optimal level of virulence for the parasite. And there's also some evidence of things like this in untreated HIV in humans as well. So this is the idea that there's some sort of optimum. Okay, so if there's some sort of optimum, that's great. Let's imagine that in the long term, there's going to be some optimum that you'll go to. But when you jump into a new host, even if there's this optimum, so we've got this graph here shows this optimal curve that potentially you have, that when you jump into your new host, you're very unlikely to be at that optimum. You could be anywhere. And then evolution will start maybe taking you slowly to that optimum. But say if you're in hypothetical phenotype B here, yeah, you start selecting to get lower virulence, always higher transmission. But if you're phenotype A, the optimum might be higher. Okay. So to some extent, that emerging pathogen could be randomly somewhere away from its optimum. So it's really hard for us to say anything about what might happen initially, whether it's going to go up or down in the long term. However, we do think we can say some things at the start of the epidemic. One of the things is I think that the characteristics of the reservoir host from which this zoonosis comes from might give us some insights into how virulent it's going to be. And Cara is going to tell us about that later and talk about how important bats are. In my own lab, what we think is very interesting is that initially, of course, when you come in to a new population, you don't go to just some sort of endemic steady state, but you have epidemics. And you remember this with COVID, a series of waves of COVID we're going through. And what we did was use models to try and predict what that does, how would the fact that you're continually in this epidemic and you come into this really susceptible population, how would that select your virulence? And we can do this by building a model. So this is a simulation of one of my computer models. This is an epidemic within the computer. It's really safe. What we have is a lot of susceptible individuals, and then we have an epidemic, goes up, then goes down, and it goes down because people become immune. And then what we're assuming is that the pathogen can then overcome this immunity by evolution, by changing, and cause an epidemic and cause these waves of epidemics that we often see. So what does this do if you have this sort of dynamic? Well, what our models tell us is that that selects for higher virulence. If you look at this graph on the right, this is what you would predict if we were at some sort of optimum, what we would assume in the model, this trade-off between how fast you transmit and how long you transmit for. But if you're in an epidemic where you're actually jumping on this huge big group of susceptibles, there's lots of them for you to infect there, then actually what you get selected for is just transmission, transmission, transmission. It's kind of a live fast and hang the consequences. Live fast, transmit quickly, and die young. And that's what's selected for. So you're really selective for being really fast at transmitting and therefore really, really high virulence. So just the fact that there's these epidemics initially will tend to select for higher virulence. And that's the sort of thing that we try and use these models to help us predict. So just to finish is, what I'm trying to say really is that, things are likely to get worse before they might get better. Okay, I'll leave you with some bees in the almonds and just to say it's first of all funded by the NSF, which is Ecology and Evolutionary Infectious Disease Program. But actually that program was shuttered last year. So we are kind of concerned how we keep funding this kind of work. Thank you.
Well, thanks, Mike. I have a question. So you showed that nice movie of the changes in infection rates. And so could I just ask, I think you may have alluded to this, but what's happening between those two spikes of infection that you showed? Is it that the virus is changing, or is this a new population of people who are susceptible, or is it a bit of both?
Yeah, so within the model, what we're assuming is that the virus is changing. So everyone gets immune, and you remember this sort of happened with COVID as well. The epidemic would happen, a lot of people get infected, immunity, it would go down, and then it can change. And so it turns into one of these new variants. And often these variants can escape the immunity to the previous variant. And that's why they take over and start dominating again. So that's what we're seeing here. We're seeing this change and this antigenic escape. You classically see that with things like flu, and I think we're still seeing it a bit with COVID-19 as well.
Well, on that question, we have one from Edwin Cheng who's asking, was the plague in the 1300s? I think that was the Black Death. Does that work with this transmission over virulence model that you propose?
Plague, transmission. I'm trying to remember what people think. We don't obviously know a great deal about the 1300 plague, but I think that it's highly virulent, it's coming across. I think that the challenge is always that when it first jumps into us, it could sort of be anywhere. We don't really know what's happened to it. And I'm not sure how much time it had to evolve in the human population. So I'm not sure how much evolution played a role in that.
Was the plague thought to be an example of a zoonosis, and where did it come from if it was a zoonosis?
I think it was from fleas, wasn't it, originally?
Flea. So it was endemic in the flea population.
Or from rodents and then transmitted by fleas, I think. Yeah.
Well, I think that may be it for the questions. I'm going to hand it back now to Mike to introduce the next speaker, Cara.
Thanks. Okay, so happy to answer more questions at the end, but now we can move on to our next speaker for the evening, which is Cara Brook, who's an assistant professor in my department of integrated biology. Brook investigates ecology and evolutionary dynamics in zoonotic disease, and particularly they focus on those from wild bats. Cara runs a really exceptional long-term study that tracks viruses in Madagascar wild fruit bats. She's really trying to understand the mechanisms that underpin transmission, how viruses are maintained in their wild hosts, and what predisposes these viruses to infect humans. She also finds novel viruses when she's doing that, which really puts her at the front line of emerging health threats. Prior to joining the faculty at Berkeley, Cara was a Miller postdoctoral fellow hosted in my lab. Cara, please tell us about your work.
Thank you, Mike, for that kind introduction. I'm going to go ahead and share my screen, and I will share my slides. As Mike mentioned, I'm Cara Brook, an assistant professor in integrative biology, and I'm going to be talking today about understanding bat virus zoonoses. So bats are natural reservoir hosts for several high-profile zoonotic viruses. These include rabies and related lyssaviruses, Ebola and Marburg filoviruses, Hendra and Nipah viruses, and of course SARS, MERS, and SARS-CoV-2 coronaviruses. My lab group has actually demonstrated that bat-borne viruses cause the highest known case fatality rates in the human population, which we show here in this graph. We have human case fatality rate on the x-axis and the number of virus species on the y-axis, and they're colored according to their reservoir host order. So these are all zoonoses. We can see that those viruses colored purple belong to order Chiroptera, or bats, and are disproportionately associated with case fatality rates in humans that exceed 90%. Intriguingly, bats appear to host these otherwise virulent viruses without themselves experiencing substantial disease, leading researchers to ask, are they somehow special in their capacity as virus hosts? Bats are special in many ways. They're the only mammals capable of powered flight, which is the most energetically costly form of terrestrial locomotion. And though we typically see an inverse correlation between high metabolic output and lifespan, bats are actually the longest lived per their body size of any mammal. I show here the log of adult body mass on the x-axis and the log of average lifespan on the y-axis, with all mammals in black and bats in red, living sometimes up to 30 or 40 years in the wild. We hypothesized that unique anti-inflammatory molecular pathways evolved to support this highly metabolic activity of flight in bats, but have then had cascading consequences on their longevity, their cancer resistance, as well as their virus tolerance. My lab group uses a combination of both empirical and theoretical techniques to understand how bat virus resistance and tolerance may actually drive the evolution of these virulent zoonoses. As we learned from Mike Boots, evolutionary theory suggests that disease results because the processes that promote pathogen transmission also cause virulence to the original host. This is highlighted in this schematic, where a virus must replicate up to some high density in some form of host excreta, shown here as a sneeze, in order to transmit to new hosts. In achieving higher densities, it elevates its transmission potential, but typically also replicates to high density inside the host, causing tissue damage that we describe as virulence. This leads to the idea of transmission virulence trade-offs and the so-called evolution of optimal virulence, whereby a pathogen evolves to maximize gains in transmission while limiting virulence costs. In the case of bat hosts that are tolerant of virus infections, we hypothesized that viruses should generate less virulence for a given level of transmission. And this leads to the prediction that viruses should evolve to maximize traits such as the within-host virus growth rate that promote transmission at limited virulence costs within the bat. Nonetheless, we would predict that these viruses would still retain their virulence, causing high pathology or high disease in non-bat hosts, including humans. Prior empirical work in my lab supports this association of bat antiviral defenses with these high growth rate viruses. I show here the fit of a spatial mathematical model to in vitro data collected in bat cell tissue culture, where we demonstrate an association of high virus growth rates with perpetually antiviral Pteropus electo bat kidney cells as compared to cells derived from other mammals, thus offering empirical support for our theoretical predictions. We use these with in-host insights into bat immunity and viral tolerance to understand the population-level process of zoonosis, the transmission of a pathogen from an animal reservoir to a human host. I show here the different barriers on the chain of events that lead up to zoonotic spillover, and we can think of these gray circles, dynamic in space and time, as mechanisms through which a pathogen can overcome these barriers to emerge into people. My lab works in a bat virus system on the island nation of Madagascar, trying to understand mechanisms across these different layers where we can intervene to reduce the risk of spillover to humans. And for today's talk, I'll focus on this level of pathogen prevalence and shedding in wild bats and demonstrate how vaccination of wild animal hosts can be used to eradicate infections prior to human exposure. My lab carries out a longitudinal field project tracking zoonotic virus dynamics in wild fruit bats in Madagascar. We work with the Malagasy NGO Association Equipa Fanihi, or Team Fruit Bat in Malagasy, which I co-founded in 2022 with a postdoc in my lab, Christian Ranaivusen. I show here some photos of our field team at work, and also highlight a few of the key members in our data collection. We work in Madagascar, the fourth largest island in the world, located off the southeastern coast of Africa, because it is home to 3 species of endemic fruit bat. Eidolon duprianum and Rosetus madagascariensis have sister species on the African continent linked to circulation and emergence of Ebola and Marburg filoviruses. Pteropus rufus belongs to the Pteropus genus that is primarily Asian in its distribution and has been historically associated with Hendra and Nipah viruses. Thus, Madagascar represents a unique phylogeographic melting pot of both African and Asian fruit bats and their respective viral clades. Bats are also consumed as a source of human food on the island, posing risks for zoonotic exposure. I show here fanihi, the Malagasy word for fruit bat, listed on a menu and being consumed on a plate at lunch. My lab undertakes capture and collection of biological samples from fruit bats at monthly time points across the island. I show here a time series of captures for our 3 different species across the different trajectories of my research career. We apply passive integrated transponders, or PIT tags, much like a microchip, to enable us to monitor individuals through time. Each line here corresponds to a single individual bat, recaptured sometimes as many as 9 times across the past 13 years of fieldwork. In the laboratory, we then process our field collected samples to build a time series of molecular and serological viral dynamics. Feces, urine, and saliva are converted to an understanding of active virus infection, and from blood, we can identify antibodies that signify a past history of virus exposure. Much of our prior molecular work required sample exportation, but our in-country Madagascar molecular capacity is continuing to expand. And in 2024, in collaboration with Brian Fisher from Cal Academy of Sciences, we inaugurated a new molecular laboratory where we now have in-country capacity for PCR sequencing and serology. Over a decade of work in this system has led us to the discovery and description of more than 60 novel bat viruses. But for today, I'll focus just on the Henipa viruses, sister species to known zoonoses derived from fruit bats, Hendra and Nipah. We can track Henipa virus dynamics in individual bats through time in order to decipher the mechanisms by which these viruses persist in wild populations. I show here a time series of Henipa virus infection status for female and male Eidolon duprianum fruit bats, where the shape color corresponds to their Henipa virus status, blue for negative and red for positive, and the lines connecting the two correspond to their background serological trajectory, or their antibody status, a past history of pathogen exposure. We can use data like these to understand the mechanisms by which these pathogens persist and also identify opportunities for intervention. And here I particularly mean, returning to this question, can we vaccinate bats to preempt future pandemics? We're currently working in collaboration with virologist Hector Aguiar-Carreño from UCLA, who has developed a multivalent Henipa virus vaccine that has 100% efficacy in a hamster model to test this vaccine in wild Eidolon dupreanum bats. From our long-term monitoring in this population, we're able to model the threshold vaccination coverage that we need to interrupt virus transmission in the wild bat population and eradicate the risk of zoonotic exposure to humans. And with that, I'll say Misocha Basica, thank you very much in Malagasy, to my lab group and my funders, and happy to take any questions.
Thank you, Cara. We've now got a few minutes for questions as well. I guess one of the key questions is always when you work on these fascinating creatures and fascinating viruses, how do you prevent getting infected yourself from one of these viruses? And
yeah, very good question. So you might have seen in the photos that we wear pretty extreme PPE or personal protective equipment. So Tyvek suits, N95 respiratory masks, double layered gloves. We have very strict protocols for sample handling, very strict protocols in the event of some kind of exposure that involve washing with soap and water, and then also povidone iodine that has virus neutralization. And then we're also very careful about the mechanisms that we use to collect our samples. So all of the samples that we bring to UC Berkeley, for example, are brought in DNA RNA shield, which is a reagent that preserves the integrity of the DNA and RNA so we can do downstream molecular work, but also neutralizes the virus, so not actually active virus. And I would say that obviously there is elevated risk in terms of contacting bats, but many of these zoonoses, as you mentioned in your talk, are not well adapted to humans and often go through an intermediate reservoir host prior to spillover into the human population, requiring some adaptation. So while there is risk, it's not the same as handling a human patient who is already infected with one of these virulent zoonoses, for example.
all right. Great. So we have some more questions from the audience as well. From Joseph, there's can you discuss the 20% vaccinated graph in more detail?
Yeah, of course. So modeling of infectious diseases in the very simplest form of transmission dynamics suggests that the proportion of the population needed to be vaccinated in order to eradicate an infection is 1 minus 1 over R naught. So R naught is the pathogen's transmissibility. So if you have an R naught of 2, then you need to vaccinate 50% of the population. Of course, this is a huge oversimplification because it assumes that we have perfect immunity following vaccination. We know that our immunity wanes after we get vaccinated for flu or for coronavirus, and it's fairly rare that we have vaccines that are actually 100% effective. And having more complex transmission cycles can also sort of muddy the waters of those estimates. But basically, we have built a model using our long-term population data of Henipa virus infections in these wild bats. We're able to fit it to those data, so replicate the data in our computer simulations. So we think we have a basic understanding of what the actual transmission dynamics of this virus are in the wild bat population. And we estimate R naught to be quite low, around 1.2, 1.1. And so with 20% vaccination coverage, using that same equation, you can actually eradicate an infection. So you've probably heard that measles, for example, we need to vaccinate 90 to 95% of the population, and that's because measles has a transmissibility of around 18. So the idea of vaccinating bats is actually fairly attainable. And I'll also note that these particular species give birth in a single annual pulse, so synchronously at one time of the year. So basically, you would need to vaccinate the population before the next birth pulse, before you get this influx of new susceptible hosts that could continue transmission.
I mean, to follow up on that, there's another question from Alan in the audience as well, which is, so that's the percentage you need to actually eradicate the disease in the bats, but would you need a lower percentage of that to actually prevent transmission or make the transmission very unlikely to the human population?
Yeah, absolutely. So vaccinating a lower percent would reduce the prevalence of the disease, or of the infection, I should say, since the bats are not necessarily experiencing pathology, which would also reduce the risk of spillover to humans. And so we've developed this vaccine in the laboratory. We're testing it in a captive population of these bats, and we're planning in the next couple of years to field test it on a small island population. Madagascar is an island, but there are also islands off the coast of Madagascar. So vaccinate a subset of the population and try to understand what the downstream implications are for pathogen circulation. I would add that partial vaccination, so incomplete vaccination of the whole population, poses some risks for virulence evolution, as you know well yourself. So that is something that we would want to consider before we were to suggest a rollout campaign that would be focused on reducing transmission as opposed to actually eradicating infection.
Yeah, and so that's actually a question from Daniel, which exactly asks that. If you vaccinate a subset of bats might you be selecting for more virulent viruses the same way as lower concentration antibiotics select for resistance? And this is something we do worry about.
Yeah, it is something that we do worry about. So, partial or incomplete vaccination, or vaccination that reduces symptoms but doesn't halt transmission, does allow the pathogen to circulate and essentially mitigates the disease burden. So, the pathogen is paying less of a virulence cost and might be selected to maximize its transmission, which then might have downstream implications for selection for higher virulence. This is something that's discussed even in human vaccines when the COVID vaccine first was released, but there are no known examples of virulence selection from imperfect vaccination in human populations, but Marek's disease, which you presented, is a great example of this actually happening in chickens. So this is a reason that we want to be very sure of our understanding of the transmission dynamics within the population before we actually take this intervention. So this project is still in fairly nascent stages, though our understanding of the pathogen circulation is fairly sound at this point.
Yeah, I think one thing to add to that, so as long as it's true, isn't it? As long as there's no transmission from vaccinated individuals, then vaccination a small proportion or a large proportion won't matter. The challenge becomes if there's still some onward transmission from those vaccinated individuals, which
Absolutely
wasn't the case with COVID-19 vaccines in humans, as far as I know. So there's a question about the peculiarities, which I think is a great question from Edwin, around what are the specific peculiarities of the immune systems that are more effective in suppressing viral infections or viral loads? Because, of course, they have the communal lifestyles and there's incredibly very dense populations. So.
Yeah. So bats are highly gregarious, as you said yourself. In fact, if you look at pan-mammalian data, the sort of average group size of bat populations is about an order of magnitude over the next closest mammal. And so certainly that means that they're under high exposure risk, and high exposure does tend to select for tolerance of viruses. Resistance becomes a bit futile when your risk of transmission or risk of encountering the pathogen is quite high. In particular, what we find in bat immune systems, and I think it's important to study bats because this opens up opportunities for exploring new therapeutic aims in humans as well. But they tend to have really reduced inflammatory responses. So they lack an entire gene, PIHIN1, that's involved in inflammatory responses in other mammals. They have really reduced reduced stimulation of this NLRP3, another gene family that's involved in inflammasome formation. And what this sort of anti-inflammatory profile allows them to do is that they can have this kind of yin and yang, where they can mount really robust virus responses, but then tamper down the immunopathology. So a lot of what we experience as disease is actually pathology from our immune response. And so bats have, I mentioned that we had this cell culture data on perpetually antiviral Pteropus electo bats. Well, they have a perpetually expressed gene, interferon alpha, that is part of the antiviral response. So they can mount these really rapid antiviral responses, but then they don't experience the sort of immunopathologic consequences as a result of that. So, and there actually is a startup based in New York City that's trying to harness features of the bat immune system for human therapeutics.
That's totally fascinating. Thanks again, Cara. We'll have some more time at the end for more questions as well. But now I'd like to turn to our final speaker of the evening. It's Tierra Smiley Evans, who's an assistant professor in integrative biology and also in the School of Public Health. The Smiley Lab studies relationship between global forest change, biodiversity loss, and disease emergence, and how we expect patterns of disease spillover impact human and wildlife health. She works on a range of mammal hosts, but she has a real focus on primates and is the chief science officer of Gorilla Doctors. Tierra holds both a PhD in infectious disease and as well as a doctorate in veterinary medicine. And I think this background gives her a unique perspective on emerging zoonosis. Tara, if you can please tell us a bit more about your work.
All right, thanks for that nice introduction. I'm going to give you a brief glimpse into what we do at the Ape Health Group. So our overarching research questions are focused on what are the key characteristics of a pandemic threat. So at the landscape scale, we think about things like anthropogenic pathogen emergence. So what are the key human activities and behaviors that lead to virus spillover from wildlife into people? At the individual organism level, we ask questions about immune biographies and adaptive priming. So how does an animal or a human's lifetime exposure to different pathogens lead to them being either more or less vulnerable to new emerging diseases? And lastly, we work with a very unique model system in the wild, the eastern gorillas. And we look at ways in which they may act as sentinels for spillover of viruses that could impact people, and also ways in which they may be uniquely resilient to disease, and what can we learn for human as well as great ape medicine. So I'd like to start us off with thinking about one key important characteristic of a pandemic threat. So we know that the vast majority of emerging pathogens in humans first originated in animals. So we conducted an evaluation of all of the known animal viruses in the published literature. And what we found was that animal viruses with higher host plasticity, meaning that they're found in a higher number and phylogenetic breadth of animal hosts, those viruses were much more likely to have animal to human spillover. And if they were to spill over, they were more likely to be amplified by human to human transmission and to have global spread. So virus host plasticity is a key characteristic of a pandemic threat. Another key overarching characteristic that we recognize is that certain species under certain conditions are more likely to have zoonotic viruses detected. So when you look at the IUCN criteria to evaluate threat and status, some key trends arise. So species that are more likely to harbor zoonotic viruses are those that are characterized as least concern with increasing population trends. So in essence, those that are likely to be flourishing in human-dominated environments. And I like to use the example of the olive baboon because they're highly prevalent in many of the field sites that I work in, and sometimes quite a nuisance. And also species with population declines due to reduction in habitat quality or exploitation. So species that are likely to be having new and intense interactions with people. So these key characteristics have really driven our research questions to focus on the forest edge. So in regions around the world where we think new pathogens are likely to emerge. And so this photograph here is actually an example of one of our field sites. This is the Bwindi Impenetrable National Park. And as you can see, you have dense agricultural landscape right up against the edge of a UNESCO World Heritage Site, which is also home to one of the highest biodiversities of primate species in the world. So as you can imagine, there's lots of opportunities for humans and wildlife to be interacting at this forage edge setting. And this is the type of landscape that we study. So I thought I would take you through a hypothetical emergence event from a forest into a human population. So this is a graphical representation of some of the dynamics of a zoonotic emergence event, and also some of the challenges that we face when trying to do this sort of landscape level surveillance. So on the left here, we typically have a wildlife reservoir host or a couple hosts, and these hosts tend to be have very low levels of viremia. And that's important because if you want to go out and do a surveillance study looking in these types of species to look for something that might be a threat to human health, it's kind of like looking for a needle in a haystack, and you're very unlikely to find interesting viruses. So next, moving along this pathway, you have wildlife reservoir hosts that are often interacting with another animal host or an intermediate host. And these animals have higher levels of viremia, greater opportunities for virus evolution, and tend to more likely be generalist species. So species that are comfortable going back and forth between forest habitats and into more human-dominated habitats where they can interact with people. Next, you have your first initial human spillover host, and these can be one or a handful of people, but importantly, they typically fall outside of typical human surveillance systems. So we often completely miss them, and they don't get studied. So next, you have your first initial human to human to human spread or human to mosquito to human spread, and this is where you have many more people involved. You can have human adapted strains, and this is where you typically first pick up there's something interesting going on and there's an outbreak. So the vast majority of infectious disease outbreak research to date is on the left side of the scale here. So our goal with our research is to move upstream. So we want to study at that optimal window for understanding adaptation and spillover, which is really focused at the forest edge. And this is exactly what we do with our research. So we design landscape gradients where we're moving from pristine forests to urban settings. And along these gradients, we conduct surveillance of longitudinal human and wildlife cohorts for pathogens focusing on arboviruses, coronaviruses, and filoviruses. So specific viral taxa that we think are the most likely to have pandemic potential. So we've been testing some of our hypotheses and landscape gradient designs in the Congo Basin forest perimeter, the Amazon Basin forest perimeter, and Myanmar karst cave systems. And I'd like to note that an important aspect of this type of work is its longitudinal nature. So we can't really conduct this research without continuing our cohort studies over longer periods of time. So we're fortunate that even though both the NIH CREED network and the NSF EEID program that Mike mentioned earlier have sunsetted or are about to sunset, we are able to continue, particularly our work in East Africa, through support of the World Bank Pandemic Fund, a capacity strengthening program. Another important aspect of our work that I'd like to highlight is actually our social science component. So we spend a lot of time thinking about how we're going to get information about key human behaviors, particularly those that may be sensitive, like hunting behaviors or contact with certain endangered wildlife. And as an example, one of our findings that I think has been quite important has been we were able to link the first connection between exposure to wildlife and exposure to Sudan virus. So we found that people who were reporting hunting primates were significantly more likely to have antibodies against Sudan virus around the windy impenetrable forest. So lastly, I'd like to talk a little bit about the opposite side of zoonoses that often goes overlooked. So we spend a lot of time thinking about what can spill over from animals into people, but I also spend a lot of time thinking about how things may go in the other direction, particularly for great apes. So there are over 140 known diseases that we share with great apes, and it's a big concern for their conservation. So I also serve as the chief scientist for an organization called the Gorilla Doctors, and we are the only organization in the world that provides hands-on veterinary care to an entire wild great ape population. This is an example photo here of our Rwandan team removing a snare from an infant gorilla. And from an epidemiological perspective, it's really an ideal system to study spillover. because eastern gorillas are 98% genetically similar to humans, and they live in three distinct geographically isolated ecosystems and are exposed to different wildlife species and in very different ways than humans interact with wildlife. So they can be looked at as a sentinel for spillover in many of these landscapes. But when people ask me what keeps you up at night in this role, it's definitely if I were ever to learn that we had a case of Ebola virus disease in an eastern gorilla. To date, that has never happened. We've never had a case, and I certainly hope it stays that way. But it is an ever-present threat. So this picture here shows you the eastern gorilla habitat in green, and the red dots represent outbreaks of Ebola virus disease in humans between 2018 and 2021. So we have had an outbreak in humans within 2 kilometers of one of the gorilla parks, and we know that wildlife reservoir hosts range within all of the gorilla parks for Ebola virus. So we've spent a lot of time together with colleagues at UC Davis and researchers around the world coming up with a plan of what would we do in the event of an index case in an eastern gorilla. And what we know through modeling exercises is that if Ebola behaves in eastern gorillas the same way it behaves in western gorillas and in chimpanzees, a single case would result in an extinction level event. So what we've also learned is that if we are able to vaccinate 50% of the population within 3 weeks, we could avoid an extinction level event. And that is what we exactly what we have prepared ourselves to do if this ever happens. But what I want to leave us with today is a bit of a cliffhanger because this is a brand new pilot study. So you guys are really the first to hear about it. But it's looking at this scenario from a different perspective. So it's very hard to explain why there has never been a case of Ebola virus disease to date in an eastern gorilla. For all practical purposes, there should have been. So this raises a very important question. Have eastern gorillas evolved an innate immunity to Ebola virus? Or are they exposed to closely related viruses that may confer some form of cross-protective humoral immunity? So to study this, we are starting a pilot project here to see if we can develop an induced pluripotent stem cell system that would allow us to study eastern gorilla immune cells easily in the lab, both here at UC Berkeley and also in our brand new molecular diagnostics laboratory in Musanze, Rwanda. And if successful, this would enable us to study viral entry mechanisms in eastern gorilla cell lines and answer some of these important questions. So I will leave us there because I know I'm at time, but I hope that you can stay tuned and continue to follow our laboratory as we grow here at UC Berkeley. Thank you very much for your time.
So we now have a few minutes for questions as well, if that's all right. I mean, maybe I can start maybe asking you the same question I asked Cara to start with, because you're dealing with things like Ebola. You know, maybe give people a sense of the protocols and the mechanisms you go through to make sure that you guys are picking up these infections yourself.
Yeah, that's a very important question. So as I said, we've never had a case in an eastern gorilla, and we hope it stays that way. But one of the important roles that gorilla doctors plays is we conduct necropsies of all dead eastern gorillas that are found within the parks and other primate species. So in the event of any dead animal in the park, we treat it as if it could have Ebola. So we wear full PPE suits, we wear, Tyvek suits, full masks, double gloves, and make sure we are taking the utmost precautions. And similarly, we make sure that any specimens that are taken for testing are inactivated in the field before they're brought back into any laboratory setting.
That's great. I guess the other question I have for you as well, you have this vaccine all ready to go. Is why don't you vaccinate them now? Does it not have a long enough? Are you worried it's going to wane or not have good protection? I was just interested in that.
Yeah, it's a good question. And it kind of hits at 2 topics. So it's a wildlife management question. There's sort of 2 camps of beliefs on how we should manage particularly wild great ape species. There's some people that think we should be completely hands-off. They should receive no veterinary care, no interactions, no vaccinations of any kind because they're truly wild and we should nature take its course. On the opposite side of that, there's people that think we should be even more proactive and be vaccinating against common childhood illnesses that are actually quite concerning for the gorillas as well. But an important thing is that we don't know how long human vaccines last in gorillas. There's no clinical trials we can do. We don't know how long antibodies last. And it's a tremendous amount of effort to try to go out and vaccinate a bunch of gorillas. Unfortunately, they don't appreciate our services as much as we would like, and it's a huge undertaking. So we would only really do it in the event of an emergency.
Okay. So there's a question from Maida here, which is about the golden monkeys that live in proximity to the eastern gorillas. Do they show evidence of diseases of note?
Yeah, the golden monkeys are a really interesting, fascinating species to go visit. They typically live in the bamboo forest zone, which is on the edge of the park. They don't really range as much into the center. So they have more opportunities to interact with people, I would say. And they're susceptible to all of the pathogens we see in the gorillas. So our biggest actually concern with human diseases that we see in the gorillas is respiratory disease. So we see a lot of human metapneumovirus, respiratory syncytial virus, common causes of human respiratory disease in people around the park.
Okay. That's great. So there's a question from Wayne who's curious. So well, you're very curious as well about the gorillas and their resistance to Ebola potentially. Have antibodies or cell level immunity been studied? Is there a known contrast with humans?
Yeah, we have done some preliminary IgG studies for exposure to filoviruses. The specificity in some of these assays are not perfect, but some of that work has been done. And the next phases are to try to see if we can look at cell entry.
That's great. Okay, thanks again, Chair. I think all these presentations have been fascinating. We have some time left, so can we invite Cara back? And I think we have some sort of questions that we could maybe all address at this point. There's a question from Edwin here, which is, whether certain perhaps people's skepticism of vaccination, how does this endanger funding for preventing epidemics? And I'm interested in your perception about whether there's been any pushback on vaccination of these wildlife animals in the same way that some people are worried about vaccination in humans. Tierra, Cara?
Sure. Yes. I mean, there's definitely been pushback about vaccination of great apes in the wild. I think it's more coming from the perspective of ethical perspective of interacting with wildlife and the stress and the management undertaking it would take to do it than it is from a perspective of some of the more concerns that we have around bats, because humans and gorillas are so similar. We expect for the most part that they would respond similarly to vaccines as opposed to other species.
Yeah.
Yeah, I can also answer. We haven't had any direct government pushback within the US. We do have both some private funding as well as some NIH funding that supports our efforts to vaccinate wild bats. That said, I do believe that there are some keywords in certain grant proposals that draw attention, and certainly vaccination is one of them. I think that some of the political drama surrounding vaccines or concerns about them is somewhat mitigated when we're not talking about actual human hosts. But that said, the Malagasy government actually has asked a lot of questions in our captive population, trying to understand if there are any risks, which I think is a very valid question on their part. They very much value their biodiversity. Madagascar has over 90% of its flora and fauna are found nowhere else else in the world. And so they just want to fully understand and be sure that that this will be a safe outcome for the species of interest. But we do have some evidence of negative fitness effects, so some morbidity consequences of virus infection in these bats. So we're hoping that it would actually help the bats rather than hurt them.
Yeah. I guess maybe I can follow up a little bit. There's a question to me from Joseph, which has said, is there any lessons in the model for someone who's never got COVID, fully vaccinated, freely out in the world now? And I guess how severe the first infection might be. I think what my model would tell you is that the vaccination is absolutely key. You're going to be very protected and much more protected than you would have been if you're unvaccinated, getting the initial virus. But getting Omicron now, the vaccine, the COVID we have now, is going to be worse than if you were getting the original one. So if you were unvaccinated now, it would be worse. So that's to say how important vaccination is in this story. There's a question about avian flu that was put in, which is a fascinating question. Maybe we have a vet here, Tierra, so I can ask you perhaps. Can you address questions around avian flu into mammals? Why isn't the mammal-to-mammal transmission in some mammals like cows and seals, but there suddenly is somewhere else? Do we have any sense about that with avian flu?
Of kind of why some mammals it transmits to and
Yeah,
while
exactly.
others. Oh gosh, it's a very good question. I don't specifically study avian flu, but I have a lot of colleagues at UC Davis who are actually currently responding to the marine mammal outbreak along the coast of California. Yeah, I don't know that I can say there's a rhyme or reason to why it's spilling into certain mammal populations versus others.
I was hoping you could, but I think you're right. And I think it goes back to this sense of how little we know about these processes, about why you suddenly get emergence, why you don't get emergence, and then what's going to happen once it comes across. It's a really big problem, I think, for the field, which is why we're excited to work in it. There's lots of questions here. I might finish with actually asking Cara what keeps her up at night. I thought Tierra said that was very interesting. So should we finish with that, Cara? What keeps you up at night?
Yeah, I think the idea of bat virus zoonosis, in particular in Madagascar. I mean, I had studied bat viruses for my PhD and postdoc prior to the COVID-19 pandemic, and it was a little bit stunning to experience this on a global scale. So I think, learning of something in our study system would be very concerning, very devastating. And there are some interventions on the human side that I didn't talk about that we're taking to try and mitigate that risk. So I showed a photo of human consumption of bats in Madagascar. We're previously had U.S. Aid funding in collaboration with Cal Academy, Brian Fisher's group, that was supporting efforts to bring insect protein into human populations in an effort to reduce wildlife consumption and lower the potential risk of zoonotic exposure. So, I think any of these actual emergence events would be the thing that scared me most.
Thank you. So I think that's all the time we have. I want to thank both speakers for being here tonight and joining me for this discussion. I'll turn it back over quickly to Dean Richard Harland for a final send-off.
Well, thank you, Mike, and a tremendous thank you also to Tierra and Cara, as well as for sharing your amazing work with us. What we've heard this evening really underscores why we call this series Basic Science Lights the Way. The challenge of zoonotic diseases is undeniably complex, as we've heard, but as we've seen also, our best defense begins with fundamental ground-level research that helps us to understand these processes. So thank you for spending your evening with us, for your excellent questions, and for your continued dedication to basic science. Your engagement and support empower our faculty and students to keep illuminating the unknown. We look forward to seeing you again next week on Wednesday, April 15th at 5 p.m. for our next event on the coevolution of Earth and its biosphere. We'll be joined by a biologist, a chemist, and an earth scientist, all studying the enormous impact life has on the chemical and physical evolution of our planet, and also vice versa. And this is becoming quite well understood in some cases. So please reach out if you'd like to learn anything more about anything we covered. As always, a video of today's session will be available on our website, basicscience.berkeley.edu, about 2 weeks from now. And you can always visit the website to see episodes that we may have missed. Until next time, stay curious, stay safe, and have a wonderful evening. Fiat Lux and go Bears.